The presentation I've asked to give is Innovations to Create New Nutritional Value from Fibre, and we've been working on this for quite a long time. And as you'll see in my presentation, we've tried a lot of different approaches. And it's not an easy nut to crack, for sure. You know, when you're dealing with a system like the rumen, which is really, if we look at, we've searched far and wide to find a system that's more important. The cellulosic ethanol guys spent billions of dollars trying to get ethanol produced from cellulose. None of them really came close to matching the efficiency of the rumen system in terms of its ability to convert fiber into metabolic products that can be used either by animals or for other purposes. And so in that case, it's pretty difficult when your task is to try to improve a system that's already pretty good. But when we talk about, really recalcitrant type feeds, which are many of the, when we talk about what we want to be feeding ruminants, and if you go to the green side of things, they say, well, we shouldn't be feeding grains to ruminants at all. We should be feeding these byproduct residues, crop residues that are being used, or byproducts of the production of food for humans. But many of those residues are even a challenge for ruminants to digest. If you're talking straws, not often the digestibility of those is less than 50%. So what approaches can we do to try to improve that and get more value out of that fiber? Well, there's been really a lot of innovations that have taken place, mostly in the molecular biology area. They've really given us the tools to look at this in a whole new manner that 15, 20 years ago was impossible to do. That just the science was not there to support the kind of work I'm going to show you today. And a lot of it revolves around, you've probably heard in the popular press, there's a lot about microbiomes. This whole microbiome thing is a great, huge area, not only in agriculture and ruminant production, but also in human health. So we have microbiomes associated with plants, microbiomes in the soil, microbiomes associated with the composting that was mentioned. Our own microbiomes, there's about 10 times more bacterial cells associated with our bodies than there is human cells. And then we have the respiratory, we heard about BRD here earlier. That's a big issue that's caused by undesirable microbiomes that can develop in the respiratory tract. And then we have the rumen, which is probably, I would argue, one of the most complex microbiomes that we've ever described, because it's composed of many different types of microorganisms, not just bacteria, but also anaerobic fungi. Less than 2 years ago, there were 6 identified species of anaerobic fungi in the rumen. Today, there's 12 that have been identified since then. So the number of anaerobic fungi that we realize that are in that ecosystem have doubled in less than 2 years. And a lot of that's because of the molecular methodologies that are enabling us to identify the unidentifiable, what was previously not identified by culturing techniques. And we also have the ciliate protozoa, and then we have the methanogens, which contribute to the methane emissions that we've heard about earlier. And these microorganisms all interact together. The protozoa even act as predators within that ecosystem. So you have an ecosystem within the rumen that's every bit as complicated as what we would have in the Serengeti plains of Africa in terms of all these organisms interacting together. Now, when it comes to fiber digestion, it's really the nature of the fiber that has an impact on how those microorganisms respond. We've had a lot of research where we talk about our own personal microbiome that each of us harbor, and there are differences between our microbiomes. But when you talk about the ruminant animal, the number one thing that dictates the nature of the microbiome they have is the type of feed that you provide to the animal. And if you alter the nature of the fiber that you provide to the animal, you'll also alter the nature of the microbiome. And that impact will be much greater than what you would see between individual variation in animals themselves. We heard a little bit this morning, and I guess there's been some stuff earlier on in the conference talking about the structure of the plant cell wall, how there's primary and secondary plant cell walls, how those differ in their digestibility, and how lignin and the linkages between lignin and hemicellulose, and how that's wrapped up in, the cellulose is wrapped up in that, has a major impact on how readily available those carbohydrates are for digestion and utilization by the rumen microorganisms. So in order to work all of this out, we've got to understand the microbial population, and we've got to understand the nature of the plant cell wall structure. We need those two things in conjunction because we need to identify the factors that are limiting the rate of digestion of the plant cell wall. If we can do that, when you look at the cellulose, if you break down cellulose, essentially what you end up with is glucose. And that glucose has exactly the same energy value as the glucose that's in starch. So if we can basically achieve the breakage of all of those linkages, we can turn fiber into grain if we want to. Now, the interaction between the microbial population and those plant cell walls is a critical component in making this all happen. And so we've done studies where we look at how that colonization takes place. And it's really a sequential process in the rumen where the bugs work together. Within 1 hour, we see the displacement of the epiphytic microbiome. That's the microbiome that's associated with the plant. Every time we feed plants to animals, we have a microbiome that we're providing to that animal that's associated with the plant. We often call those organisms to be transient. Typically, they die in the rumen because the rumen's an unfavorable environment for much of the plant microbiome, or that members of the plant microbiome, like some of the fungi we heard about in the talk this morning, actually move and transfer right through. They're transient. They move through the rumen and end up in the feces. Then after the displacement of that, you have the primary colonizers that start to attach and start to produce enzymes that digest the feed. And eventually you get secondary colonization where you get a very complex, what we call a microbial consortia or a biofilm. Where that community is working together to carry out the digestion of the plant cell wall. Now the amount of enzymes, it takes many enzymes to break down the plant cell wall. No one microorganism in itself produces all the enzymes that are capable of doing that. So that's why the microbes have to work together as a team in order to break down that fiber. So this is just sort of a cartoon that I published quite a long time ago that sort of illustrates that. How you have the primary colonizers. They're the first ones that attach, and we can see them. They'll use these carbohydrates they call glycocalyx to attach to the plant cell wall. And then they'll attract these secondary colonizers that you see here. And those secondary colonizers actually use the end products of the primary colonizers. So the primary colonizers produce waste products as part of their metabolism. The secondary colonizers use those waste products as their metabolites. And they break those down further, and eventually we get the VFAs produced. It's the VFAs that the animal ends up using. So you have a continuous stream there that helps accelerate the whole process. As the animal absorbs VFAs, it stops the VFAs from inhibiting the secondary colonizers. As the secondary colonizers use the substrates produced by the first primary colonizers, it also prevents them from being inhibited. So it accelerates the whole process in terms of the digestion. That's why the rumen is so efficient at digesting fiber. And we can look at the electron microscope and see these microorganisms attaching to the surface. This is corn stover. And you can see how they form chains, and you can see how they duplicate and how they start to form pits. That's because they're generating enzymes which are excreting, often 360 degrees from the cell surface. So you end up with the erosion of the plant cell wall in the vicinity of the cell itself. And that became more and more complex. This is actually a confocal laser micrograph shot. These are protozoa that you see here, these circular structures. That's actually their nucleus that you see. And the blue little dots you see on the surface there, those are methanogens. The methanogens and the protozoa actually work together because one of the end products of the activity of the protozoa is hydrogen. And they have an organelle called the hydrogenosome, which the methanogens love because the methanogens need hydrogen as a substrate to produce methane. So they hang out close to the protozoa, attaching, a lot like you'll see if you ever watch the Blue Planet, where you've got the fish swimming in the sea and the one that's attached to the other guy all the time, waiting for morsels of food to fall off. Well, this is a lot similar. The methanogens are sitting there waiting for the hydrogen that's being produced by the protozoa. And the same thing, that methanogen then will also help remove that hydrogen end product and accelerate the digestion process at the expense of producing methane. Now, really, I mentioned it's the omics that's really give us these new novel strategies that we can use. And it's the ability for us to take a digestive sample, to extract the DNA or the RNA from that sample, sequence it, and then run it through a bioinformatic pipeline where we basically describe all the genes that are present within that sample. If we do it on RNA, then we're dealing with the genes that are being transcribed. They have the opportunity to be produced into protein. Not all genes are transcribed. So a gene that's not transcribed does not produce a protein. And from all of that bioinformatic analysis, we can start to determine who's there. So we use a gene called 16S RNA. And by looking at that sequence, we can tell which bacteria that represents. Or we can tell what they're doing by what kind of genes are they producing that are being transcribed. And then we can start to predict the function. So that's how we've started to apply that then towards fiber digestion. So basically, just from the gene alone, you can then predict the enzyme structure. You can then expect which substrates that enzyme is going to interact with and which end products it's going to end up producing as a result of that interaction. All on the basis of just the gene itself. We can make those kinds of predictions now. So it really opened up the avenue in terms of the broadness and the types of enzymes and the diversity of enzymes that we're actually able to study. Now, the colonization, these bacteria work together as a team, but they also have varying roles. So this just shows the colonization by bacteria. So we're using that gene 16S to look at the colonization, in this case of fiber, over a 48-hour period. And you can see how Prevotella predominates earlier on, but other bacteria come in to play a role later on in the digestion process. So that sequence is really important. And then by basically sequencing those bacteria, looking for the genes that they have within their genome, we can then understand what enzymes might become available as these other microorganisms or other bacteria colonize later in the digestive process. So then you can start to develop a visual model about how the microbes work together to carry out that fiber digestion and the type of enzymes that they're bringing in later on in that process to achieve the digestion of the fiber itself. Now, there's a huge database, which is called the KZ database, and it represents all of the carbohydrate-associated digesting enzymes. And right now, there's about 300 families of different enzymes. In that, about 300,000 different enzymes have been described, a little over 300,000. And some of those are involved directly in the digestion of the bonds between the various sugars or various carbohydrates. And some of them are involved in the cleavage of the linkages between lignin or other decorative compounds like acetyl groups and that, which will cleave those linkages from the fiber as well. And then some are also just involved in binding of the enzyme to the fiber itself. There has to be some degree of association in order for the enzyme to digest the fiber. Now, we can go into the rumen of various animals, which has been done. In this case, the musk oxen, Holstein cow, Angus cow, Jersey cow, and even the yak, and even the wallaby work that was done out of Australia. Many labs have done this kind of work with different types of herbivores. And you can see then we can sequence all of that messenger RNA and look at the kinds of casezymes that are being produced. So they just classify these with numbers. So each of these represents a different family. And when we look at the types of families we see, you can see that some we see are quite prominent in ruminant animals, like the GH5s, are in most of the cases are being detected. But you can see some of them are completely absent, like the GS7s. GH42s, 44 is not very common. GH12, GH62. So we can start to identify those casezymes that appear to be absent from the rumen, raising the question, if we were to find those enzymes in other environments and introduce them into the rumen, could we be adding enzymes that are some of those rate-limiting enzymes that the microbial population is not producing that are required to further enhance that fiber digestion process? So the next step then is to start to search other environments for those kinds of enzymes, and that's what we've been doing. So we've went into a lot of wild ruminants like elk, white-tailed deer, moose. We worked with the plains bison. I showed a little bit of the data on musk oxen, mule deer as well. And we've even started to deal with some of the rodents because of the types of very recalcitrant fiber sources they consume, like the beaver and the porcupine. And I'll look at the relationship relative to the types of casezymes that we see in cattle. When we look at the microbial populations across those different types of herbivores, you can see that we see quite dramatic differences in the nature of those populations. We don't have to get into the weeds about what all of these different bacterial represent, but just if you look at the colors, you can see that in the wild ruminants, we tend to have a greater diversity than we do in domesticated. In the case of the bison, these were farm bison, not wild bison. So you can see they're actually quite close to what we see with the cow, with the bovine. And then we have very different patterns with the rodents between the beaver and the porcupine. So again, I think this is really reflecting that the type of feed that these animals are consuming is driving the nature of the microbial population. So when you're dealing with wild ruminants, where they typically have a more diverse diet, they're eating both grasses and browse and sedges, etc., it has a more diverse population relative to the population that you're dealing with in farm domesticated ruminants. And when we look at those distributions, like in this case, the porcupine and the beaver, we have different distributions. There's much higher levels of casezymes associated with the digestion of pectin and hemicellulose in beavers and porcupines relative to the amount of xylan. We find much higher levels of xylan in ruminant animals. So would supplementation with some of these enzymes offer benefit to the ruminant animal? That's the question. And these porcupines will eat some pretty rough-looking stuff. So this is just a shot that we took. We have a lot of porcupines actually around Lethbridge, and this is what they eat during the winter. They'll just clear the bark right off of those trees. So we have to think about where the sources are coming from, but we also have to think about the microbial population itself, because we cannot afford to compromise the efficiency of the microbial population at the expense of any additive that we put in there. It has to complement, it has to be synergistic, not to replace it. And one of the issues we have is there's a lot of work that's been done with probiotics, but invariably most of that work is showing that unless you inoculate the animal every single day, that microbe does not persist within that rumen environment because it's not competitive. So in this study, what we wanted to try to gain a handle on is if we take microbes that are not been growing in a really nice environment, like a big 1,000-liter fermenter where all other competition was wiped out through the use of antibiotics or whatever means of specific substrate to prevent other microbes from colonizing within that fermenter, and they also grow in those conditions with unlimited feed source. There's no competition for feed with other microorganisms within that environment. Can we dramatically change the nature of that rumen environment? So how we did that study is we looked at the transfer from, and there's work about bison being more efficient at fiber digestion than cattle. So we looked at transferring contents, digestive contents from bison into cattle. In that case, those microbes are in the competitive environment within the bison, and they're being transferred into a competitive environment within the cow. And we transferred 70% of the rumen contents of bison into the cattle. So we took 70% of the contents from the cattle out and put in 70% bison contents to see how much we could change that microbial population, and could we improve digestion based on the evidence there is that bison are more efficient at using lower quality fiber. Now, we used really low quality fiber in this study. We fed these animals 75% barley straw, so they weren't getting a lot of energy and very high in fiber. And this is basically how we did it. We slaughtered the bison. We didn't have windows in the bison. We only had windows in the cows. Transferred the contents immediately into a central system. Used a Canadian tool like the paddle, to keep it Canadian with the canoe paddle, to mix things up. This was under anaerobic conditions because we had CO2 flowing into this system. And then transfer through the window of each individual cow 70% of those contents. And then look at the impact that that had on digestibility and the change in the population. So when we can do a procedure called principal component analysis, which really shows where the populations cluster. And you can see that this is the cattle population on day one before we did any transfer. This is the cattle population, of the bison before we did any transfer. This is the cattle population before transfer. 1 day of transfer switched the cattle population to here. And then 27 days after the population was transferred, it was at this point. So even after 27 days, the bison population had still had an impact in changing the nature of the microbiome in the cattle. In a lot of studies, they found that it reverts back to being the cattle population after a period of time. Paul Weimer did some work showing that when he transferred between individual cattle, but not when he transferred with bison directly. When we look at the impact on fiber digestion, this is total tract fiber digestion and NDF digestibility. We found over a 10% range between the highest and the lowest animal. But after the transfer, we didn't find really that any animal was a dramatic improvement. There was still a range from 43 to 56 even after that transfer. So it doesn't look like the transfer of those contents really had a big positive effect on fiber digestion. I think that might have been because those were farmed bison. They weren't bison that were out grazing the plains. And the quality of the diet, when we looked at it, was actually better than what the cattle were getting in the barley straw diet. So probably those microbes in the bison were not adapted specifically to the digestion of that fiber. Again, emphasizing that it's primarily the nature of the microbes that dictate the fiber digestion. And when we look between low and high efficiency animals, we find that there are some differences in the types of casezymes that are present, but nothing jumps out as being dramatically different. And it actually looks like, Lou Guan up at the University of Alberta has done some work saying that it might be related more to having less diversity of these casezymes being more efficient in terms of fiber digestion relative to more diversity. Now, how can we take all that information and boil it down into something that we might be able to use to develop an enzyme preparation? Well, we can use the proteomics. I showed how we could use that to document and estimate the type of protein that might exist. We can use FTIR for cell wall analysis, so we characterize the nature of the plant cell wall. We can incubate microorganisms with those plant cell walls and look to see what kind of genes are expressed in order to digest those. And then we can go ahead and also look at direct plant cell wall modification as well. We run that through a system of screening procedures that we use, but we do it in light of the impact on the enzymes that are already being produced in the cow itself. So the enzymes that procedures we're developing now take into consideration the nature of the enzymes that are being produced by the cow as well. And we do that by taking contents from low and high digesters, running that through a freeze-drying process to lyophilize it, and basically get an enzyme preparation that originated from the cow itself. And then we have a system, what we call a microassay, where we can produce microgram quantities of enzyme. One of the issues that's been passed with enzyme screening is that in the other procedures it takes quite a bit of enzyme, recombinant enzyme, in order to test the value of that enzyme. This assay allows us to do that with microgram quantities, so we can produce those enzymes in E. coli in the laboratory and then test their effectiveness when combined with the rumen enzymes for that synergy that I was talking about. And basically we just run that assay through and look at glucose and sugar release in combination with those rumen enzymes with our candidate enzyme of choice. Once we identify candidate enzymes that look favorable, we can then scale up the production of those individual protein or enzymes and look at it in the batch culture, which everybody I think is pretty familiar with in terms of how that works. And then also take that and look at it in the artificial rumen system where you have a continuous fermentation system. So that's the system we use to run and select enzymes that are showing promise. And we've done that in a variety of different ways. So this is an example of where we took and basically cloned a gene coding for one of those esterases that break the linkages between ferulic acid and hemicellulose and from the rumen fungi, Anaeromyces. We produced that. Now, the other challenge is that you've got so many combinations of mixtures. If you have one enzyme and you add it in with rumen mixed enzyme, or you add a mixture of enzymes, how do you select which mixture is the best in terms of its activity? And we used a program to try to predict that called Design Expert Software, where you basically run a large number of variations of the mixtures that you're trying to formulate. And based on that, you look at the release of carbohydrate or glucose or sugars from those various mixtures. And that program will identify the optimal combination. So in this case, you can see that as we adjust the various amounts of those mixtures that we're adding into this system, we end up with the highest amount of glucose release associated with this system here. And in that selection, it actually eliminated this enzyme even from being in the mixture. So we've used that to design cocktails that enhance the release of sugars in the presence of rumen mixed enzymes. And we've used that same approach then also. So here's the activity of the rumen mixed enzymes. And here's a bunch of candidates that we're screening for their ability to enhance the activity of sugar release from those mixed enzymes. And you can see this particular candidate looked very promising. And you can do that with mixtures of enzymes as well, or individual recombinant at the same time. You can also look at what is the active protein or enzyme that's having the impact within mixtures of enzymes. So in this case, we fractionate the enzyme mixture on a Blue Native page. We can then cut each of these enzymes out of the gel itself, resuspend it, incubate it through our microassay with mixed rumen enzymes, and identify which of those bands is responsible for the greatest increase in sugar release in that screening process. So that starts to then tell us which enzymes are most important. And then we can go back to the predictive software and predict which other enzymes do we already have in our collection that should exhibit similar properties to that one that we found in the mixture that's responsible for the improvement. Now, the other approach is to actually, when we talk about the exogenous enzymes that are out there now, most of those preparations increase the rate of fiber digestion. They don't increase the extent of fiber digestion. So there's more fiber digested per unit of time, but the total amount of fiber that's digested doesn't change very much at all. In this work, what we're trying to do is get at that indigestible fiber. The stuff that we saw today, I think, was 240-hour incubation period. So basically what we've done is we've collected that undigestible NDF, and then ran it through a similar process of screening like I've just described, but using it as the substrate rather than just barley straw. At the same time, then we use some immunolocalization techniques where basically we use that to describe the nature of that plant cell wall that's indigestible. That helps us then target our enzymes towards those indigestible components. And we can use FTIR to also further characterize that indigestible material. And when we do the enzyme fingerprinting for that, for barley straw, relative to what we see for those indigestible residues, the types of enzymes formulations we come up with differ between those substrates, because there's quite a bit of difference between the indigestible residue relative to the barley straw itself. So that's kind of what we've been doing on the enzyme front. We've also taken some studies looking at the plant side. So that same ferulic acid esterase that I talked about earlier, we took that same gene and we actually expressed it directly in alfalfa. So we made transgenic alfalfa that produced that enzyme. And we basically looked at expressing that in 4 different plant tissues: the apoplast, chloroplast, endoplasmic reticulum, and the vacuole. And these spots here just show that that enzyme expression is taking place after the transgenic procedure. When we look at the ability for that to increase the liberation or solubility of lignin within alfalfa, we found that 2 of those tended to increase that above the control. Most of them increased it to some extent, but not all statistically significant. But it didn't alter the cell wall composition in terms of its carbohydrate profile. So it's not changing the composition of the hemicellulose cellulose directly. All it's doing is changing the degree of linkage because of that enzyme that the alfalfa is producing. And when we looked at the glucose release in some of those lines relative to the control, we also see increased release of glucose when we incubate that with commercial enzyme preparations. So it makes sense that that ferulic acid esterase is breaking those linkages to lignin when we have good expression within the plant, which is not in all cases. And then that when you expose that to fibrolytic enzymes, it increases the release of the glucose from those plant cell walls. The issues around that is, one, it's a transgenic plant, and the acceptance of transgenic plants, although there's lots out there already, there hasn't been a lot of new ones being released. So that's one of the issues. And the other is, of course, you can't compromise the yield or productivity of the plant in any way as a result of this transgenic trait. And we never got to the stage of field trials to know whether that was the case or not. And then finally, I just want to finish with a little bit of a different approach where you look at a chemical treatment. The treatment we've been working with just recently, in the last couple of years actually, was developed by MBI at Michigan. They have a pilot plant for AFEX production in Lansing, Michigan. And they basically contacted us because we were looking at interactions between that and enzyme treatment as well. Basically, the process is just a matter of grinding the 4-H, putting it in a chamber, adding in steam and heat, introducing ammonia vapor, running it through a soaking procedure, depressurizing it, and then stripping that back off and getting a final product that's then pelleted. They went ahead and produced that for wheat straw and rice straw for us and shipped it up to Canada, and we ran some trials with it. And in the trials we did, the plant is not huge, so to produce this is actually quite laborious. And we only did studies in sheep because we couldn't produce enough material for cattle. But basically what we did in our studies, these are lambs, where we took diets, balanced them out, and took alfalfa pellets and basically replaced 30% of the alfalfa with wheat straw directly in the diet. The rest of the dietary components are the same. And when you look at digestibility, you can see that we improved the digestibility of NDF and ADF in those diets as a result of the AFEX treatment directly. And we also, if you look at dry matter intake, was higher on the AFEX treated material. So we substituted out 30% alfalfa, put in 30% wheat straw, and we still get higher intakes with the AFEX treatment. And when you look at the average gain to feed was a little bit lower for the AFEX material, but when you consider that grade of level, adding 30% wheat straw to lamb diets is usually not a good idea. We still had a real marginal difference in gain to feed. So I think it really shows that this treatment can dramatically improve the value of wheat straw as a fiber source from an energy perspective. And we've done some additional work looking at this, and it also enhances the activity of the enzymes as well. So the enzymes can make some improvement on control untreated wheat straw, but if we add them with the AFEX material, the improvement's even better. So in conclusion, the keys to enhancing ruminal fiber digestion, we need to have the right microbes at the right time, and we want to make sure those microbes are working efficiently to carry out those processes I described. Any exogenous enzyme should be synergistic, not neutral, or antagonistic to the rumen enzymes that are naturally produced. And I think the unique thing about sort of the strategy we're taking now is that we're trying to select those enzymes in light of their interaction with the rumen enzymes. Most of the past commercial enzymes that were out and evaluated and assessed in the feed industry were originally developed for some other purpose. They were not developed specifically for ruminants. And this is, and we do see some positive responses to those at sometimes, but I'd say the big issue with those preparations is that the response is unpredictable. And we're trying to put predictability into it by considering the rumen enzymes as part of the equation. We need to understand the physical and chemical natures of the plant cell wall structure, so we make sure we have the right enzymes to target those right rate-limiting steps. We can use chemical destruction of plant cell wall to sort of complement that. There are issues when it talks about transporting forages long distances and that, and central pilot plants or plants at an industrial scale to produce that on a significant scale. There are challenges around that. I believe MBI now is primarily doing a lot of work in India and China because they have the huge issue over there with regard to rice straw and the impact that has on air quality because they burn a lot of that material. And if you've ever been in those countries when they're doing that, it's a horrible experience. So basically, that's a lot of, and they're getting funding from the Bill Gates Foundation, so I think that's where Bill Gates is also focusing his efforts. And then the plant cell wall carbohydrates, remember that they are all sugars, and if we can make those sugars available and liberate the energy from them, they'll have the same energy value as the other feeds that we're currently using to provide energy in the diet. So with that, I just want to thank the team back home that helps with all the research we do and answer any questions if I have time. Thank you.
Thank you very much, Dr. McAllister. Any question? We have got plenty of time for questions. We've got two mics, one with me and one with Bruce at the end.
Yes, thank you for your presentation. You mentioned the work on enzymes and on the plant side. Would it be, is there any way to think about colonizing the rumen of a newborn calf with some combination of bugs?
Yep. So the work that's been done, I mentioned with probiotics and how we've done some of that work ourselves, where we've added microbes into the rumen directly, and we find using tracking techniques, we can track those individual organisms now. They last about 9 hours after they're introduced into the rumen. Where we see the greatest potential is where that level of competition is the lowest. And that level of competition is the lowest in the newborn animal because that microbiome is not fully established yet at that point. And actually, we do have some grants in that we're putting in right now to do some early life studies. And there's been some work done in the UK as well. Jamie Newbold's group's done some work where they've looked at that from the perspective of methane emissions, where they have basically colonized young lambs with microbes that lower methane emissions, and they see that those methane emissions do remain lower in later life as well. So that's where the most potential is for those exactly. Fiber digestion is a little bit harder because the young newborn is not fully up to speed with fiber digestion instantaneously either. The level of complexity there creates a bit of a challenge. But whether we can achieve that or not is something certainly worth exploring.
Do you do any work looking at the microbiome from the soil to the plant all the way to the rumen?
Yeah, we've done some work on that in relation to silage production. So we've looked at the nature of the microbiome on the plant while it's growing out in the field and how that infects the ensiling process. And the ensiling process is complex as well from a microbiome perspective because you have major shifts going on in the ensiling process. And likewise, when you take that silage out of the pit, you have a change in that microbiome again. We do find, like, if you've got issues, with contamination, like the discussion this morning when we were talking about mycotoxins and fungi, if you have soil material, we can detect that in the silage at the time of ensiling. if you've been making that mistake by running your header too low and bringing dirt up into the system. We haven't specifically looked at how the nature of the microbiome in the soil affects the plant's health. That's getting a little further away from my area that's usually somehow connected to beef in one way or the other.
How do things are in your development process where you have hemicellulose-cellulose ratios that change and then the linkages to the core lignin? Do you need a different enzyme cocktail, or maybe you said it and I missed it?
No, I didn't say anything about that, but that's a really good question. In some ways, I'm reluctant to answer that question because it introduces increased complexity in this whole process, right? Because we know that adding enzymes, I think most producers find adding enzymes to the diet to be a bit of a pain in the ass in terms of having to do that. If they see major performance responses, they're probably prepared to actually go ahead and do it. But if you tell them, okay, you're going to change from now from alfalfa silage to corn silage in your diet, you've got to change your enzyme up now because your fiber source has changed from a monocot to a dicot or those kinds of changes.
Well, that's what
then they're going to become less enthusiastic about that, right?
Yeah, because we, in the dairy sector, we have such a range in from legumes to grasses and that type of thing, and I've often wondered if...
Some of the work that we have done so far would suggest that, yeah, that might be required. I guess the trick would be, can we find those rate-limiting enzymes that have implications for multiple forage types? And could we use those as a way of simplifying that issue? Yeah.
And do just as a follow-up a little, do the fungi play a role in helping in the microbiome community and enhancing fiber digestibility?
Yeah. So the rumen fungi are actually associated with the digestion of the most recalcitrant fiber. In the past, people kind of downplayed the rumen fungi because they only account for about 8% of the total microbial biomass in the rumen. But when we started doing these studies where we're measuring which genes are being expressed that have an impact on fiber digestion, in some studies we're seeing that 60 to 70% of those enzymes are coming from the rumen fungi. So even though you can't use the size of the biomass as an indicator of the relative importance of those microbes within the rumen, they all have their role. And even though they're present in smaller numbers, like 8% that I said, versus bacteria or protozoa that can be 50% of the biomass, they still are playing a big role. And that you can have a small amount of biomass with a lot of enzyme expression. And that's what we think we're seeing with the rumen fungi.
Any more questions? Okay, with that, thank you, Dr. McAllister.
Thanks a lot.
Thank you.